Hello and welcome to Chapter 7. Today we're going to be talking about DNA replication, transcription, translation, and a little bit of gene regulation. So let's go ahead and get started. So just a quick overview, the diversity of living organisms is really due to genetic diversity, so differences in the genome. And what's really kind of fascinating about genetics is we have 4 bases, right? Think about it, A, T, C, and G. So we have just 4 bases, and these 4 molecules make up the blueprint for all of the living things on our planet. Everything living, even some things that are not living but still pathogenic, such as viruses, which are not considered living organisms, but viruses are genetic material. So everything out there that is considered living or, again, non-living uses this genetic material, this A, T, C, and G, to determine function and what we call a genotype and phenotype. Now, DNA itself is a really elegant molecule. It's a very simple structure. Most DNA is linear, although in bacteria we're going to be talking mostly about prokaryotes, and prokaryotic DNA is circular. It is made with those 4 molecules, A, T, C, and G, and it works in a very similar manner to binary code, which is used in computers. So think about everything that our computers can do. I'm just thinking about even just my desktop computer here that I'm looking at. It can do some really amazing things. And if I think about what my computer can do with binary code, right, with just ones and zeros, what could I possibly do if I just doubled that capacity? And in DNA, that's what we're doing. We're doubling that capacity of a binary code because we're using 4 bases instead of 2. And so the opportunities in that are pretty endless. Now, the genome of a cell is the complete DNA and or RNA sequences that are found in a cell. And the proteome is determined. The proteome is all of the proteins within a cell. And what proteins are expressed in a cell is determined by the genome. So the proteome is determined by the genome. A gene itself is a specific DNA sequence within the genome that encodes for something. Most are structural genes. A structural gene encodes for a protein. We have non-structural genes, which encode for things like ribozymes, RNAs, those sorts of things that are non-protein. So a gene, again, a gene is a very specific sequence. So it's a small region within an entire genome that encodes for something. And there are 2 basic cellular functions that are required of DNA that are important for DNA to be a part of. One is replication. Genetic material has to be replicated because every cell needs to have a copy of it. But in addition to that, we have expression of that genetic material. And expression is really important for cellular function. So these are the 2 required cell functions of DNA, requirements of that particular material. Now let's take a look at some structure and some characteristics of the genetic material itself. DNA, we know, is double-stranded, and it is in a helical form because of how the bases are put together in what we call a sugar phosphate backbone. That's what we're looking at here. So in that sugar phosphate backbone, the way that our bases are stacked, it causes our DNA molecule to be twisted in this helical form. We have what's called a 5' carbon, a 3' carbon, and a nitrogenous base. And this is important in the actual structure of a nucleotide. So I'm just going to do a little rough drawing here. So I have a oxygen, right? And I'm looking at my base here. And sticking off of here is my, say, adenine or guanine, whatever base it may be. And then I have a carbon up here, and then I have some phosphates. So this is your typical nucleotide here. And in organic chemistry, you can see it's very similar to what we're looking at in this picture here. In organic chemistry, carbons are numbered in molecular structures when we draw these molecules. The actual carbons in the molecules are numbered. And in our bases, adenine, thymine, guanine, cytosine, these are what are called purines and pyrimidines. They have ring structures as well as the actual sugar itself. So here's my sugar, and here's my base, and these bases are quite large and they're made with ring structures as well. And the carbons in organic chemistry, when we draw these structures, we give each carbon in the molecule a number. But in nucleotides, we need to differentiate between the carbons that are associated with the bases, with the A, T, C, and G, and the carbons that are associated with the deoxyribose sugar that makes up the DNA molecule. So the bases are numbered the carbons on the bases are 1, 2, 3, 4, 5, 6, 7, 8, etc. But the carbons that make up the sugar, the deoxyribose sugar, are given the designation prime. And when we write that, it's written as 1 prime, a little apostrophe there, right? And then we would have 2 prime and 3 prime. And when we number these sugars on our base here, we would see we move to the right. So this is carbon 1, carbon 2 prime. This is 3 prime. This is 4 prime. And then this carbon up here is actually 5 prime. So again, I'll just number it on my drawing down here. I have carbon 1 prime, 2 prime, 3 prime, 4 prime, and up here this is 5 prime. And this is an important distinction because this 3 prime, although I can't draw it, it won't draw here on my thing. I have a little OH down here that's coming off of a little, yeah, OH, you can see it here, right? This OH coming off of my 3 prime carbon there. And this structure is important because when DNA is put together, what you possibly can hopefully see here is that when DNA is put together, the 3 prime carbon of this deoxyribose sugar binds to the same phosphate group as the 5 prime carbon of the neighboring sugar. That leaves this 3 prime carbon open to bind with the 5 prime on this one, leaving the 3 prime to bind to the 5, the 3 to bind to the 5, and then this is the end of my molecule. And since this 3 prime carbon here is not bound to an incoming sugar, to another sugar, again, that's the end of the molecule, this is referred to as the 3 prime end of this DNA sequence. Just like at the top here, you can see this 5 prime carbon is bound to this phosphate. That 5 prime phosphate is free. It's not bound to anything else. So this is the 5 prime end of my strand of DNA. And in order for my bases, for the bases to fit together and complementary base pair like they should in DNA, my second strand has to be parallel. But in order for the bases to face the other strand, to face inwards and be in the correct orientation for binding, not only does it need to be parallel, but it has to be parallel in the opposite direction. So it's referred to as anti-parallel. And so I want you to just kind of take your hands, right, and let's pretend that your thumb is the 5 prime carbon and your pinky, right, your pinky is the 3 prime carbon. Okay, your pinkies are the 3 prime carbon. And these 3 fingers, or the bottom of your hand, let's do it this way. The bottom of your hand is the 3 prime carbon. And so we need our bases to base pair together. And if I'm parallel, but my the ends of my 5 prime ends are both at the top, my bases are not going to come into contact with each other to bind. Now, I could make this upside down like that, but then my bases in one of my strands is facing in the wrong direction. So in order to get my bases to be able to bind, I not only have to be parallel, but I have to be anti-parallel in the opposite direction. So by flipping it over like this, I have 5 prime here, 5 prime here. My 3 primes are in the opposite direction, but my bases are now together, and my bases are able to bind because they're now in the correct orientation. So whenever you're confused about directionality, just make your thumb a 5 prime carbon at the end of a DNA sequence and take a look at how you can make your bases bind. And once you get your bases bind, you can see the structure of a DNA molecule. Now the bases, when bases bind to each other, they are complementary. So A will always bind to T and C will always bind to G. Now just to kind of show you some of the elegance of a DNA molecule, when A binds to T, it will do so with 2 hydrogen bonds, and when G binds to C, it does so with 3 hydrogen bonds. You can see that here. They're showing 2 here and 3 here. Regions of DNA that are encoded, that are transcribed, we should say constitutively, meaning they're constantly needed. These are genes that are going to be used all the time. Those genes in the very beginning, where those genes need to, where the DNA needs to open up so that transcription can occur on that gene, that area is going to be what we call a TA-rich region. And that region in front of that gene has a whole bunch of TA. It's actually referred to as a Tata box. And that TA-rich region is because when A binds to T, it's only 2 hydrogen bonds. Fewer hydrogen bonds means it's easier for the enzymes to get in there to begin that transcription process. Regions of the genome in which they might be permanently turned off, or they're not transcribed very easily. We want to really restrict that gene as much as possible. They have what are called CPG islands. And CPG islands are CG-rich or GC-rich regions. And these GC-rich regions are where we have a whole bunch of cytosines and guanines all complementary base paired together. So we would have a big long sequence of C's and G's. And that's because that's 3 hydrogen bonds. And when we have a lot of hydrogen bonds collectively, it makes our molecule very strong. So this anti-parallel property, remember, anti-parallel, complementary base pairing, hydrogen bonds in the center, the bases put together, the deoxyribose sugars, creating the sugar phosphate backbone, is put together by covalent bonds. In particular, it's called a phosphoester bond or phosphodiester bond, and they are covalent bonds. So the sequence of amino acids is maintained. That integrity is maintained through the strength of millions of covalent bonds, and then the 2 strands are held together but flexible for reading because the 2 strands are held together by hydrogen bonds. So RNA is another piece of genetic material. RNA is single-stranded. It uses a ribose sugar instead of a deoxyribose sugar. So where we had an oxygen missing, right, where we had just a hydrogen in our deoxyribose, in a ribose sugar, we have an OH group. So instead of the hydrogen being bound there, it's bound to the oxygen here, and it's a hydroxyl. In the base pairing of RNA, we have A binding to U instead of G, and we have C binding to G. So it uses the same nucleotides, it just replaces thymine with U. So if you see a genetic sequence and it has a bunch of U's in it, that just simply means that it is RNA instead of DNA. We have 3 primary functional forms of RNA. There's a few other odds and ends out there, but for the most part, we're looking at ribosomal RNA, transfer RNA, and messenger RNA. And all 3 of these RNAs are involved in transcription and transfer and or translation. I'm sorry, this is translation. Not transformation. That should be transcription and translation. Over here, we have just a quick little GIF, and I just was trying to show that all 3 of these types of RNA are really, all 3 together, what help to carry out the process of translation, which is protein synthesis. So the little green guys coming in, these are tRNAs. This right here is our mRNA. And then bound inside of the actual ribosome here, which is in blue, is our ribosomal RNA. And it's part of that ribosome. This red line coming up out of the top, that's the actual polypeptide that's being synthesized in this particular picture. Bacteria also have to be able to regulate their expression of their genes. All living organisms have some form of genetic regulation, and this is because a cell is not going to just simply produce express every gene it has all at the same time. Sometimes a gene is necessary, sometimes it is not. It depends on the cellular function, the environment in which the cell is located, and what that cell's interaction is with possibly with other cells, like a population or something like that. So DNA replication. So let's start with our first primary function of DNA in the cell, and that is DNA replication. DNA replication will begin at a start sequence. And we're going to look at a plasmid. Plasmids in bacteria are very small circular pieces of DNA in addition to their chromosome. However, this process that we're looking at applies to the chromosome as well. And in bacteria, most bacteria, their chromosomes are circular like a plasmid. Just plasmids are smaller, a little easier to handle. So it has an origin of replication. This is a DNA sequence that is recognized by the machinery known as a replisome. The machinery necessary, it's enzymatic machinery for replication, to actually carry out the process of replication. And when replication occurs, this machinery is going to hop in there and it's going to start to divide the two strands from each other. These are referred to as the parent strands or template strands. So the two templates separate from each other and create what's known as a replication fork. And the replication fork is just the region in which replication is actually occurring. And it's called a fork because it's just separating these two strands of DNA from each other. The replication forks will continue. So replication, like you can see in this diagram, replication is going to continue, and it is bidirectional. It's going in two directions, right? The opposite directions at the same time. So it's referred to as bidirectional. And we have a bidirectional replication until it gets down here at the bottom. And at the bottom, there's going to be another DNA sequence that is referred to as a termination sequence. And this is where it says, hey, look, we've met, we're done. And the two replication machineries come together. They replicate that final end, and the whole complex falls apart. When that complex falls apart, we now have two copies, right? So here's copy 1 and here's copy 2 of that genetic material. This process is known as initiation, elongation, and termination. You're going to see these three terms for DNA replication, transcription, and translation. All three of them will go through these three stages of process: initiation, elongation, and termination. So let's talk about the process itself and what actually happens during DNA replication. So here we have an origin of replication. Right? We're at an origin of replication. We have two replication forks, one on the left, this region here, and one on the right. The very first thing that happens, we're going to look at replication moving towards the left first, okay, just to take a look at it. So we'll just concentrate on the left side of this picture. The first step, step number 1, is DNA gyrase and helicase. These are two enzymes that come together to help form what's called the replisome. Helicase is an enzyme that is responsible for separating the DNA strands. So it kind of works like a little bullnose, and it kind of just pushes in between the two strands, wedges in between the two strands, creating the replication fork. And what it's doing is it's just cleaving those hydrogen bonds between the bases to help separate the two strands so we can get our replication machinery in there. DNA gyrase is actually found up here, just in front of helicase. Still part of the process, part of that replisome complex, but it's found in front of helicase where the DNA is still double-stranded. Because DNA is helical, as helicase moves through, it's going to cause over-twisting of the DNA in front of the replication fork. So DNA gyrase comes in and just kind of relaxes the DNA and allows it to untwist while helicase is pushing up against it. So it's releasing torque or torsion that may be occurring on the DNA. This is important because we need to make sure that the DNA does not break. And if we put too much torque on the DNA, we twist it too much, the DNA will eventually break and we won't be able to complete the replication process. Next, we add some single-stranded binding proteins. Those would kind of fit right on the ends here of these nucleotides as they're being separated by helicase. These are important to make sure that these bases do not innocuously bind with anything else. We don't want them to self-bind, so we don't want the DNA to get bent in on itself. We don't want them to just kind of randomly bind with nucleotides that may be floating around in the environment, because this is all occurring in the cytoplasm of a bacterial cell, so we don't want them to just kind of randomly bind. And we certainly don't want it to zip back up together and re-base pair perhaps in the wrong order, in the wrong place. So it all needs to be kind of protected. So these single-stranded binding proteins just kind of act like little, right? They're going to be like little covers on those nucleotides just to protect them, and it's very temporary. The next step is primase, right? So this was step 2, and now we're in step 3. In step 3, an enzyme called a primase is going to come along and it's going to attach somewhere up here and it's going to lay down this primer. Right, so I'm just going to draw it in here so it's a little easier to see. So here we have our primer and this is primase. Now, the reason we need a primer, and the primer, by the way, is made out of RNA nucleotides. And the primase uses RNA nucleotides so that other proofreading enzymes can come back later and recognize it and say, hey, that's RNA, we need to replace that. And that's going to happen later. But primase lays down a primer because our next step, step 4, DNA polymerase 3, when it synthesizes a new DNA strand, it can only add an incoming strand's 5' carbon right here to an already existing 3' carbon, which is what the primer is leaving behind. And this directionality is important in DNA polymerase 3. DNA polymerase 3 is replicating, and it can only replicate in one direction. And so it's going to replicate from the 5' end towards the 3' end. And it's needing here, it has to have an existing 3' carbon available for the incoming nucleotide. And when I show you this video at the bottom here, you will see that. So the primer is used to provide an existing 3' carbon so that DNA polymerase 3 can begin this process of synthesis. It has to have something to attach to get started. Which brings us to step 4, which is DNA polymerase 3. Now DNA polymerase 3 synthesizes this new strand and it's moving along here. Remember, we're going to the left, so it's moving along the 3 to 5 strand, but it's replicating in a 5 to 3 direction. Once DNA polymerase 3 has begun its replication, and we're looking at this top strand up here, and this is called leading strand synthesis. And we're looking at this top strand. Let me get that up here. DNA polymerase 3, once it starts, it just follows the direction of the replication fork, right? We're moving to the left. It's going to follow the direction of the replication fork, and it's called continuous replication. So this is continuous. And this continuous replication, this polymerase, this is polymerase 3, right? So we'll just put a big 3 here. It's going to follow that replication fork. It's continuous. It's one big long replication strand. But now that polymerase 3 is moving with the replication fork, now I have a problem. I still have an RNA primer over here. So I need to come in with another polymerase. In this case, it's DNA polymerase 1. And DNA polymerase 1 is going to replace the RNA primer that primase laid down and replace it with DNA nucleotides. So instead of it being ribose sugars, it's going to be deoxyribose sugars. But again, DNA polymerase has its own limitations, and it cannot create that bond between 3 to 5 and 5 to 3. So DNA ligase comes in and repairs that. So where that little nick will be between the new DNA nucleotides from DNA polymerase 1 and the existing strands, it's going to come in and it's going to fix that. So DNA ligase works on the sugar phosphate backbone. It does not move nucleotides around. It just simply glues everything together. Now you'll notice in DNA ligase we have the term Okazaki fragment. And this refers to what we call lagging strand synthesis, which is that bottom strand. Now we go through all of the same steps and in the same order. But the problem is because of that directionality of DNA polymerase, here my replication is going to be moving away from the replication fork. So this means if I started, say, over here, right? If I started, here's my primer, I'm going to replicate in that direction, but my replication fork is moving in the other direction, right? My replication fork is moving to the left here. So I need to find a way to lay down another primer over here and then carry out replication as usual. And then eventually when these strands here separate, I'm going to lay down another primer up here and then that one's going to move across. And what will happen is DNA polymerase 3 will replicate just as it did on the leading strand. It's just going to do it in these small fragments. And my DNA polymerase 3 will move along replicating in this direction until it runs into the primer from the fragment in front of it. When it hits this primer, the polymerase goes, well, that's already been replicated. I don't need to replicate that. It falls apart. And it goes back over here. It'll fall off here, go back over here, and start the process again with a new primer. Meanwhile, polymerase 1 will come in and remove the primer, replace it with DNA, and then DNA ligase will come in, join those fragments together, and we end up with one big long DNA molecule. So it's all done. In a replication fork, notice it's bidirectional replication, so we have movement to the right just like we had movement to the left. But because we are moving in the opposite direction, now my top strand becomes my lagging strand and my bottom strand becomes my leading. And again, this is because of the directionality of DNA in combination with the limitation of DNA polymerase 3 only being able to synthesize in the 5 to 3' direction. I'm going to show you this video real quick and see if this kind of helps you gather a visual.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So hopefully that video was helpful to you in kind of getting an image and seeing how replication occurs and how the directionality of DNA polymerase 3 plays a role in that leading versus lagging strand synthesis. There we go. So this video here talks a lot about the structure of DNA and its relation to some replication.
Do you recognize this molecule? This is DNA, or deoxyribonucleic acid. By the end of this video, you will be able to identify the key structural features of DNA, as well as describe the importance of those features for function. During this video, we will look at different representations of the DNA molecule to better view certain details, but all views represent this same structure. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix, which is what we will discuss here, is also called B-form DNA. Now, let's move to a more simplified representation of DNA to discuss the details. We can unwind the double helix like this, so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon, which is present in ribose. Because of this, nucleotides in DNA, deoxyribonucleic acid, are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left, and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3', and the bottom strand orientation 3' to 5'. These strands are also sometimes called Watson and Crick. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT, or TA, and GC, or CG, base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the base is attached to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms two repeating and alternating spaces called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks. This is the DNA double helix, and you've now learned the structural features that influence its function. We hope you've enjoyed exploring this amazing molecule with us.
So as you can see, although DNA seems really simple, there's a lot of complexity to it. And what's really kind of cool about it is that simple structure allows for all of this complex use and its role and how it is used within the cell to carry out cellular functions. Come on. There we go. So this is just a drawing I had to kind of review with you DNA replication and the process again. So remember, we have step 1 where we are going to add helicase, right? So helicase is in front here with DNA gyrase up here. DNA gyrase is an enzyme that's in the class of enzymes called topoisomerases. You will see the term topoisomerase when they talk about eukaryotic DNA replication. So DNA gyrase is bacterial topoisomerase. So helicase, again, that's going to break these hydrogen bonds, leaving a whole bunch of unbound bases behind. I'll just put some bases in here for you. And in our next step, we're going to lay down a primer. So I'm going to just come over here. Here we have our primer. I'm just going to write primer here. And that's going to leave, that primer is going to leave a 3' carbon here, this 3' carbon unbound. It's ready. They're base paired, but the RNA base, the RNA sugar, is not. And so remember, this is an RNA primer. In my next step, DNA polymerase 3, right? So I'll just put DNA polymerase 3 comes in, and it's going to start adding nucleotides. Now, this nucleotide here that it's added, this is its 5' end, right? So it's doing it so it can base pair, and it can pair up, create that sugar phosphate backbone with an existing 3' carbon. It's going to then replicate, and it's going to move towards the replication fork. Once DNA polymerase begins creating, right, so it's moving this way and it's making all of these bases here, and it's creating this form, this new strand of DNA, we're now going to move on to our next step, which is our DNA polymerase 1. Like in the video, an exonuclease works with DNA polymerase 1, and it removes the primer and replaces it with DNA molecules. So we'll just make it a little darker here to symbolize that. Replaces that primer with DNA, so we're going to call that polymerase 1. And then DNA ligase will come in and repair the sugar phosphate backbone. I know this is very messy. You can draw your own, use different colors, that makes it helpful. Lagging strand synthesis is the exact same step. So I have helicase and gyrase. Well, of course, remember we had our single-stranded binding proteins on here. I forgot that in the first one. There you go. I'm going to add my primer. And instead of moving with the replication fork and lagging strand, I have to move away from it. Right? This is my 3' carbon. And DNA polymerase can only add to a 3' carbon. So my polymerase is going to move in that direction. So that means that up here I need to add another primer. Right? So here's another primer. And my DNA polymerase 3 is going to move in that direction. And when this polymerase runs into this existing primer, it'll fall off. Then we follow all the same steps that we followed in leading. So my primer gets replaced with DNA nucleotides by polymerase 1. And then DNA ligase comes in and repairs this sugar phosphate backbone so that these Okazaki fragments, that's what they're called, Okazaki fragments, can all be joined together to give us one continuous copy. And so in blue, those are the parent strands, and the daughter strands are in red. And this is also why it's considered a semi-conservative process. It's conservative in that we maintain the sequence of the DNA nucleotides within those parent strands. They become a part of the copy, but it's semi-conservative because half of it is new, and the other half is from the previous strand. And then in the next replication round, both of these strands here will become parent strands again. So that's our lecture on DNA replication. I'm going to end it here. It's kind of a long one, but DNA replication is a complex process, and I wanted to make sure that we reviewed it several times in a few different ways to find what way works best for you. Just so you know, one of the best ways to really understand or be able to visualize DNA replication is to actually draw the process on a piece of paper. Draw it out the same way over and over and over again. Make sure that you label 3' to 5' in the replication fork, and then draw each of those steps over and over again for both leading and lagging synthesis. Because lagging synthesis can be a little confusing when you're first learning it, and if you realize that it's just simply leading synthesis over and over and over again in small pieces in the opposite direction, the steps will become second nature to you, and you'll be able to actually visualize how that's occurring on the DNA strand. So our next lecture will be on gene expression, and we will talk about transcription and translation. Have a good day. See you in class.